invitation. This is my first time in person here. It's a great event, so happy to be here and to share with you today some data on new NAD precursor, Trigonelline, with muscle longevity benefits across species. So actually, by looking at the structure of Trigonelline, what emerges is that nothing more than a methylated form of nicotinic acid, or niacin, which is a known NAD precursor. It has also been, let's say, around or known to the people for a long time already for its anti-diabetic effects, essentially in the context of several mouse models to ameliorate metabolic syndrome. What's also important to know is that it's a molecule that is present in our bodies, but is actually largely produced in plants, such as fenugreek seeds, coffee beans, and so on. And in fact, actually, when you drink coffee, you will most likely get a spike of trigonelline in your plasma. Also, there is very early evidence from the 80s that people have tried to do biochemical characterization for whether there could be a possibility to demethylate trigonelline, and seems like the liver may be able to do so and to generate nicotinic acid. So with this in mind, and having measured trigonelline in the serum of patients, before doing experiments, we wanted to really just do a very simple experiment, and there are two interesting things to notice here. So we wanted to test the stability of trigonelline in human serum. So we just spiked trigonelline at 1 millimolar at 37 degrees in serum. And we took along actually the two of the most famous NAD precursors currently on the market. Also, you have seen the talk from Morten about NR and NMN. And we looked at the stability in serum. So trigonelline is very, very stable, and that's interesting for us, and that's good to know. However, this is something that is known, and it's not a critic or anything. It's just to raise the awareness that although the NAD precursors, they do work, they do come with some complications. And especially for clinical translation, this is very relevant to keep in mind. So, for instance, in RNNM, at 2 hours, they already significant decline in serum, and at 24 hours, they are largely gone. And if you look here at their structure, essentially it's they resemble each other, and in fact, you can see that, for instance, they release abundantly NAM when present in the serum. So again, this is just to keep in mind that for clinical translation, it's very important to measure and look at the NAD metabolome when we provide precursors. You have seen in the beautiful talks already all throughout these days that aging is basically characterized by several dysfunctional marks that occur across different tissues, and about possible interventions to target the aging hallmarks. What's also important to know that in the context of muscle aging, some of these hallmarks are also shared with sarcopenia, which is the loss of muscle function and mass indeed associated with aging. From a clinical perspective, there are several worldwide aligned guidelines on how to define sarcopenia, and there are essentially three main parameters used as indicators of muscle health: gait speed, grip strength, and appendicular lean mass, which I will also use in the next slide, so keep these three parameters in mind. So recent work indeed before I joined Nestlé Research was trying to focus on how to characterize further looking at muscle biopsies sarcopenic hallmarks. But they did so by looking at several cohorts scattered across the globe, and in particular here I'm showing the sort of clinical features of a Singaporean cohort, where you can see here the three clinical readouts of muscle health. And what they did by essentially looking at these two cohorts and looking at muscle biopsies, on one end is they found that at the transcriptomics level and proteomics level, downregulation mitochondria function. However, what also emerged very clearly is that in the muscle of sarcopenic patients, NAD levels were declined. And importantly, although the amount of samples here of muscle biopsies was low, NAD levels were positively correlated with the three indicators of muscle health, so with gait speed, grip strength, and appendicular lean mass. Now, with this in mind, when I joined and we launched the project as a follow-up to further characterize now what could be systemic factors or molecules linked to what we have seen, especially considering the muscle levels of NAD were declined, we made use of serum samples from the same Singaporean cohort from which we analyzed the muscle biopsies. And we tried to look at, for instance, metabolites that may be linked, for instance, to NAD, in particular vitamin B3-related metabolites. And looking at this, the only one that was significantly changed, and in particular downregulated in the sarcopenic cohort, was this metabolite called trigonelline. Importantly, as for the NAD muscle, trigonelline levels in the serum also positively correlated with lean mass, grip strength, and walking speed. And also very interestingly, when we looked at how the serum trigonelline levels were associating in a positive manner with pathways in the muscle biopsies, what emerged by far is that the most positively enriched pathway was the OXPHOS-related genes. You can see here the enrichment on the enrichment score plot, and also here the genes that are enriched in the particular gene set. We started with the human primary myotube cells, shown here, where we show that trigonelline can dose-dependently increase NAD levels, and it actually even works in primary myotubes derived from sarcopenic patients, indicating that actually it's possible to have a response to trigonelline even if you may have sarcopenia. We then switched to C. elegans as a short-lived animal model, and we saw that trigonelline can increase NAD levels or restore NAD levels to a more youthful state. And when we gavaged adult young mice with trigonelline for 2 hours, we observed a consistent increase in NAD levels across several tissues. Next, we wanted actually to really map the conversion route of trigonelline into the NAD biosynthesis pathway. And here in this cartoon here, there are some of the pathways that also Evandro and Torren Finkel have mentioned. For instance, you have the prior-secondary pathway that makes use of nicotinic acid through the MPRT enzyme to produce NAD. And you have the salvage pathway, which uses NAM and basically, or the NRK pathway through NR to generate NMN and NAD. And this, basically, the key enzyme here is NMPT, which you may have heard of before. So what we did here, we also leveraged the use of inhibitors such as FK66 for NMPT and 2-hydroxynicotinic acid that is essentially an inhibitor for MPRT. And then we test this in the with or without supplementing the precursors. So what you see here in control conditions in myotubes, when you supplement either NR or trigonelline, you get a nice increase in NAD levels. However, when you now co-treat with 2-hydroxynicotinic acid to block MPRT, trigonelline doesn't work anymore. And furthermore, when we now combine the two inhibitors and we do extended experiments to induce NAD depletion with FK66, which is known to occur in several cell types with this inhibitor, you can see that trigonelline and NR, they can nicely rescue NAD levels and even boost them further with NR, indicating that indeed trigonelline does not seem to rely on the salvage pathway. But when we now combine the two inhibitors, trigonelline effects are completely abolished, really supporting the idea that it makes use, like the nicotinic acid, of the prior-secondary pathway. Next, we wanted to couple the same experimental design that I mentioned you before for the NAD levels, and keeping in mind the use of the inhibitors for the two pathways here, looking at mitochondrial function. In particular, we looked at mitochondrial membrane potential and maximal mitochondrial respiration. Again, due to NAD depletion, now in this case, you see that the mitochondrial respiration readouts are declined, but are promptly restored by either trigonelline and NR to comparable manners. But when we now combine FK66 with 2-hydroxynicotinic acid, trigonelline is not able to restore mitochondrial function as well anymore, compared to NR that instead just works just fine because it can bypass the two pathways. Next, we wanted actually to test the benefits, since that was our interest from the beginning, on muscle aging of trigonelline. We started again with the C. elegans due to the fact that it's a short-lived animal, and we treated the worms from day one of adulthood actually to bypass any potential effect on development. When we looked at muscle fiber integrity, trigonelline was able to actually ameliorate the muscle aging phenotype, and this was mirrored by actually an improvement in the mobility of the worms, because you see less paralysis here in blue in the worms over time compared to the control condition. And very importantly, trigonelline was also able to extend lifespan, but it was doing so relying basically on the expression of MPRT1, which is the worm ortholog for MPRT mammals. So indicating that there is an evolutionary conservation of the utilization of trigonelline for also muscle health benefits that requires the prior-secondary pathway. Finally, we wanted actually to go back to mammalians, and to do so, we made use of 2-year-old mice, supplemented chronically with trigonelline for 12 weeks. And at the end of the study, after supplementation, we measured grip strength and in situ muscle force. This is a protocol essentially where you take a muscle of interest. In our case, we use the TA muscle, and then you force through electrical stimulations, repeated contractions of the muscle to induce muscle fatigue. So from this long-term treatment, we got sort of two kind of sets of phenotypes, if you want. On one end, the lean mass was not impacted, and the peak force, so the maximal force that the mouse can do when you force the muscle to contract, was not changed. However, the grip strength in these mice there was a small but significant rescue, and this was also mirrored by an improvement in the muscle fatigue phenotype. You can see here that when you start the repeated contractions in the muscle of the aged mice here in black, there is a steep decline in the amount of force that they can generate versus the young mice here in gray, which recover much easier in that sense. And here with Trigonelline, we sort of partially rescue this phenotype, showing here this intermediate phenotype here in blue. So there is, of course, much more that we could discuss, but time is short. So I want to conclude by acknowledging all the people that helped us with their expertise.